Hello to everybody watching. I just wanted to take a sec here at the beginning to make clear that I am not a professional, okay? What you're about to watch, is just my stand-up comedy routine. So, enjoy. Okay, alright, welcome to class everybody. Today we are talking about research designs, the methods and ethics of research for neuroscience, Chapter 4. Starting out with science, research, and theory. Alright, researchers in psychological science conduct studies with one of three goals in mind. 1. Describing behaviors. 2. Examining the relationships between behaviors. And 3. Examining how different variables impact behaviors. Now, which question the researcher wants to answer will ultimately determine which research method they choose to use. The three different designs most heavily used in behavioral neuroscience are case studies, correlational research, and experiments. That is what we're getting into. Case studies, correlational research, and experiments. So, starting off with case studies. A case study is just an examination of a single person or group in detail over a period of time. Like, for example, studying a patient who is missing their hippocampus. Case study is the primary descriptive research design used in behavioral neuroscience. In fact, case studies have been used historically in this field. Case studies provide researchers with a ton of detail about a specific person or condition, and they can give researchers insight that lets them develop broader studies. But case studies do have their downsides. For one, they typically focus on a person who has a rare characteristic or rare characteristics that make it hard to generalize or apply any findings to other people. Right? So because case studies tell us a lot about extraordinary individuals or extraordinary groups, the research, the findings from the case study is difficult to generalize to larger, more ordinary populations. Correlational research is a methodology that examines the strength and direction of a relationship between variables. If we want to look beyond just describing behaviors and want to actually know about the relationships between them, then we would use correlational research. For example, the relationship between study time and grades. Correlational research can be conducted using some descriptive research strategies for collecting data, but the difference in this case is that the researchers are measuring two variables and then using them to calculate a statistical component known as the correlation coefficient. The correlation coefficient is just a value between -1 and +1 that represents the strength and direction of the relationship between variables. A positive value for the correlation coefficient means that the two variables are positively correlated. As the value of one variable increases, so does the value of the other variable. For example, age and height are positively correlated in children. As kids get older, we expect them also to get taller. It is a positive correlation, positive value of the correlation coefficient. On the flip side, a negative value means that the two variables are negatively correlated, right? So as the value of one variable increases, the value of the other variable decreases. Example, as a person gets older, their agility declines. So in this example, we see an increase in age, but a decrease in agility. As age increases, agility decreases. That is a negative correlation. Oh, and I should also mention that with the positive value, right, so the positive value of the correlation coefficient, two variables go up together, but they also go down together, right? So if I have two variables with positive correlation, they're both going up, and they both go down, right? They go together. With a negative correlation, right, what that means is that as one goes up, the other goes down. As one goes down, the other goes up. They are negatively correlated, negative value of the correlation coefficient. The closer the correlation coefficient is to either -1 or +1, the stronger the relationship between the variables is. A correlation coefficient of 0 indicates that no relationship exists between the variables. So to kind of explain this, right, so, one second. I am a professional. Okay, so let me make sure I have this straight. Okay, so here's +1, here's -1, all right? The closer the correlation coefficient is to +1, the stronger positive correlation we have on our hands, right? So the closer it is to +1, the stronger positive correlation we have, right? -1. The closer the correlation coefficient is to -1, the stronger a negative correlation we have, right? So imagine this little spectrum here, okay? So if we're sitting really tight over here, we got us a strong negative correlation. If we're sitting real tight over here, we got us a strong positive correlation. Remember, positive correlation, the variables go together, right? So as one increases, the other also increases. As one decreases, the other also decreases. But over here, all right, negative correlation, as one variable increases, the other variable decreases. And as one variable decreases, the other variable increases, right? So one goes up, the other goes down. Whereas over here, one goes up, the other goes up. If one goes down, the other goes down, right? Now on this spectrum, in between them, is 0, okay? That's a 0. The closer we are to 0, the weaker the relationship is, all right? And at 0, there is no relationship. So if we're closer to here, it's a stronger positive correlation. If we're closer over here, it is a stronger negative correlation. But the closer we are to 0, the weaker the relationship is, be it positive or negative. And at 0, there is no relationship, all right? It is so weak, it's non-existent, all right? Anything at 0, nah. Correlation coefficient of 0 means no relationship. A correlation coefficient of 0 indicates that no relationship exists between the variables. Now, an important thing to note here is that correlation does not tell us anything about causation. Or, as we put it in class, correlation does not imply causation. And if you want to look up something to that point, look up, I believe this website is Spurious Correlations. Correlation is not causation. All right, good, got that point. Okie dokie, the third and final research design that we're going to be talking about are the experiments. Experiments focus on the relationship between independent and dependent variables. To say anything about causality, we need to use experimental research, which requires things like random assignment of participants to experimental and control conditions, and it allows us to draw conclusions about the causes of events. In the field of neuroscience, we can actually do true experiments. I'll explain why in a minute. Experimental studies are much cleaner when we want to understand causation, but they're harder to do. So if you want to poke at causality, you need to do experiments, and they're going to be damn hard to do. We're talking about manipulating the variables of interest, right? That is what makes it difficult, and the manipulation can also be ethically questionable. Experimental studies are powerful in that they give us answers about causality, but they are difficult to carry out and they are limited by ethics, just to really drive that point home. And like I said, we can do true experiments in behavioral neuroscience thanks to improvements in technology. A good example of this, we randomly assign people to different levels of brain function, all right? And then we manipulate brain function in a safe manner to see what aspect of the brain causes certain changes in behavior. By manipulating brain function, all right, we're able to get a measure on what certain functions each part of the brain may be responsible for. That is an experiment. The independent variable is the variable being changed or manipulated by a researcher, whereas the dependent variable is the variable being measured by a researcher. The dependent variable depends on the manipulation. It depends on the independent variable. So again, remember, we manipulate the independent variable, all right, and we measure the dependent variable. So let's say, for example, we are testing the effectiveness of medication on stopping smoking. In this case, the independent variable would be whether the participants get the medication, all right, because we, as the researchers, control that. while the dependent variable would be how many cigarettes they smoked before and after they actually took the medication, because that depends on the independent variable, the treatment in this case. The experimental group is the group containing participants who receive treatment or who participate in some experimental condition, whereas the control group is the group containing participants who do not receive treatment or who do not participate in the experimental condition. To make sure that the experiment is accurate, researchers use random assignment to determine which participants will be placed into experimental or control groups. Random assignment is a process in which each participant has an equal chance of being placed into either the experimental or the control group. Why do we do this? Well, random assignment is done to minimize the impacts of confounding variables. So if you get a question on the exam like, why do we use random assignment? The answer is we do it to minimize the impacts of confounding variables. Let's talk about confounding variables. Confounding variables are uncontrolled factors that a researcher does not account for in a study, but that could potentially influence study outcomes. So basically, confounding variables are the things that you did not plan for, things that you did not anticipate, things that you did not control for, that may still come around to influence your results, thereby really hurting how good your study is. To finish off with the experimental stuff, I just want to note that the high level of control in experiments allows researchers to draw conclusions about cause and effect. But because experiments are so controlled, they may not mirror the real situation that they're trying to study, right? So the lab may be too artificial. What we do in the lab may not reflect the real world because of how controlled it is. All right, let's talk about what they have in common. All three designs require that researchers come up with operational definitions of the variables that they want to study. An operational definition is the specific description of how a variable will be measured as determined by a researcher. Operational definition is important because many questions that we may have about behavior are generally too vague or too open-ended to be useful to researchers. So instead of working with a bunch of loosely defined concepts, we instead use an operational definition to really tighten the ship on what we're talking about and make sure that we have a specific description of how a variable will be measured as per what the researcher says. To this point, instead of measuring something like sleep deprivation, we would instead say that we, as the researchers, are going to measure how many participants get less than 4 hours of sleep per night, or something like that. In this case, the loosey-goosey definition of sleep deprivation has been refined and specified to be anything less than 4 hours of sleep per night. That's basically your operational definition. In short, all right, you must be clear about what definition you are picking so that other researchers know exactly what your study measured. It just really helps with communication, because if I do one thing in my study and somebody else wants to do the same thing in their own separate study, it would really help them out if I had a good operational definition. All right, that's research designs. That's all the research designs. Remember, research designs we talked about include the case study, correlational study, and the experimental stuff. Now, we're getting into research techniques. All right, so here we're talking about staining, stereotaxic surgery, and brain imaging. Yeah. Let's talk about staining techniques first. All right. Staining techniques allow researchers to study nerve cell anatomy and take measurements. All right. So with staining techniques, you can actually see the anatomy of nerve cells using these stains. A good example of this would be if we wanted to investigate the effects of weed on mice brains. So what we would do is we would let the mice blaze it up, then we would kill them, we would slice into their brains and apply a bunch of stain to the dead 420 mice brains, and then, lo and behold, we can actually see how wacky their brains have become after having smoked it up. Now, staining techniques are old, but they're still pretty cool because they allow us to see the organization of nerve cells, like the nerve cells in a person with schizophrenia. So let's talk about these stains. We have the Golgi stains, the myelin stains, the Nissl stains, and there's a little thing about the Ch.4 stain also that I'm going to throw in there. Focus on the Golgi stains first. Golgi stains highlight individual neurons. Golgi stains allow us to see nerve cells. We emphasize the Golgi staining because staining nerve cells was a huge breakthrough that allowed us to see the structure of nerve cells. So the shorthand for this, Golgi stains highlight individual neurons. Myelin stains then emphasize white matter and therefore also emphasize neural pathways. Those pathways get stained blue. Then we have Nissl stains. Nissl stains emphasize the cell bodies of neurons, right? These bodies get stained dark. They stain the dendrites and the somas, or the gray matter. And last but not least, Ch.4 stains mark the nuclei of active neurons. The Ch.4 detection is a cellular technique that indicates global changes in activity in order to analyze neuronal populations involved in response to, well, several stimuli. Ch.4 expression might be used as a marker for neural activity throughout the neuraxis following peripheral stimulation. Now, those are a lot of words. Don't worry, all you really need to remember is that Ch.4 stains mark the nuclei of active neurons. Nissl stains emphasize the cell bodies of neurons. They stain dendrites and somas, or gray matter. Myelin stains emphasize white matter, and therefore also emphasize neural pathways. Myelin stains stain the pathways blue. And, of course, Golgi stains, they highlight individual neurons. They let us see nerve cells. They were a huge breakthrough. Real quick before we move on, I want to talk about immunocytochemistry, because immunocytochemistry uses antibodies attached to a dye to identify cellular components, like a receptor, neurotransmitters, or enzymes. So, in short, biggest takeaway from this is that we do have techniques to go down to the level of nerve cells. We can create biochemicals that bind to the proteins within nerve cells, and so this will allow us to see what is going on inside. Now let's talk about stereotaxic surgery. An old piece of technology is a stereotaxic instrument, and it is used to place an electrode or some other device at a precise location in the brain. A stereotaxic instrument allows you to lower electrodes into very precise areas of the brain. Now, there are stereotaxic devices for humans, but they are not used for research. You will not catch any researcher using a stereotaxic instrument on somebody just for the sake of research alone. Stereotaxic instruments are only used on humans for surgery. All right, we're talking surgical procedures. Now, granted, if somebody just so happens to be getting a surgery done and they have one of these machines working on them, then sure, a researcher may come around and be like, hey, do you want to be part of a case study? They would study the patient before and after the surgery. But again, you would not use a stereotaxic instrument for the sake of research alone. At least when it comes to humans. All right. The stereotaxic instrument is used in animal research. All right, so when it comes to research on animals, yeah, then you can use a stereotaxic instrument. But when it comes to humans, we only use stereotaxic instruments for medical conditions. All right, we're talking surgeries. The important thing to note here is that stereotaxic surgery is a blunt force technique to study the brain. If you don't know what function a certain part of the brain serves, you can knock that area out and see which function has been inhibited. We essentially disrupt a part of the brain and see what changes in the person. And again, I gotta really hammer this point home. Stereotaxic surgery is not done for research on humans, but if a single individual has a surgery done, then they may become part of a case study. A little part of this that I want to note also before moving on is the stereotaxic atlas. All right, the stereotaxic atlas shows the location of key structures on images of a series of brain sections. So you have your stereotaxic instrument, and then you use your stereotaxic atlas to see the location of key structures on images of whatever brain sections you're looking at. All right. Finally, brain imaging techniques. Brain imaging techniques that we talk about include EEG, PET, fMRI, There's a little bit on there on MRI. And then I also want to talk about TMS. All right, but let's back it up. Focusing first on EEG. Behavioral neuroscience emphasizes brain imaging technology. The EEG stands for electroencephalograph. It sums the electrical activity of neurons between two electrodes. The EEG detects fast-changing brain activity, but it is very poor at localizing it. The EEG operates within a time span of 1 millisecond, and it analyzes a spatial span of 10 to 15 millimeters. The EEG measures the electrical activity of the brain. The measurement occurs on the center of the scalp. With EEG, we have electrodes that are embedded in an EEG head cap, and these can pick up brain activity. The electrodes detect the ions moving in and out of nerve cells. Now, EEG does not do a very good job of telling us where activity is occurring, but EEG is good for picking up very rapid changes in the brain, and people can be easily trained to use EEG, so it is accessible and it is used extensively because it is so easy to get. It's not very good at telling you where activity is occurring, but it is really good at picking up rapid activity. So it'll tell you, hey, something's popping off, it's going really fast, but I can't really tell you where it is. That's EEG. Then we have PET, positron emission tomography. This is an image produced by emissions from injected substances that have been made radioactive. PET tracks changing activity. It detects receptors, all that good stuff. PET operates within a time span of 30 seconds, which is a relatively long time, but it analyzes a spatial span of 2 to 3 millimeters. When a particular area of the brain becomes more active, blood flow increases to that part of the brain. So what researchers will do is they will inject a small amount of radioactive material into a person, and then this material will go with the flow of blood to whatever area of the brain is most active at the moment that person does something, which a researcher can then look for to see what part of the brain is active when that person is doing something. Now remember, with PET, it takes 30 seconds to detect the radioactive material, which makes PET useful for detecting which part of the brain is active for long, sustained activity. So we're not talking about like an activity like snapping your fingers. Happens too quickly. We're talking about long, sustained activities, right? Something that you can do for at least 30 seconds, because 30 seconds is how long it takes for the researcher to detect the radioactive material, which remember has been going with the flow to the part of the brain that is being activated when you're doing some odd, long, sustained activity. So, with all that in mind, PET is not useful for detecting rapid change, but PET can be used to detect specific areas that are active, because remember, very specific areas. We're talking 2 to 3 millimeters. That is very small. In a lot of ways, PET is the opposite of EEG, because EEG could tell you that really rapid things were happening, but it couldn't tell you where they were really occurring, because EEG has a spatial span of like 10 to 15 millimeters. But then PET can tell you where things are happening because it has a spatial span of 2 to 3 millimeters, so like really precise, but it takes its sweet time to get detected, so you wouldn't use PET for anything rapid. You would have to use PET for something long and sustained. Oh, and also when it comes to PET, you gotta be very careful because you are using radioactive material, so there's that. Another big one that I want to talk about is fMRI. All right, so fMRI stands for Functional Magnetic Resonance Imaging. fMRI detects increases in oxygen levels during neural activity. It tracks changing activity. fMRI operates within a time span of 1 second, and it analyzes a spatial span of 1 to 2 millimeters. Now, if you compare this to EEG and PET, you'll see that fMRI is way better than both of them. It is way more accurate in its spatial span, and it is way faster. All right, we're talking 1 second operating time, and it analyzes a spatial span of like 1 to 2 millimeters. That is so freaking tiny. fMRI is the gold standard of brain imaging techniques. This is overall the best brain imaging technology that we have right now. However, its downside is that fMRI is very technologically intensive, and it's also really expensive, so you gotta train people on it, and it's gonna really blow out the bank. But again, fMRI can track rapid changes in the brain, and it is really good at determining where activity occurs. And to kind of compare this with an ordinary MRI sees structure alone. All right, so an ordinary MRI is just structure, whereas the fMRI sees both structure and activity. MRI, just structure. fMRI, that's structure and activity. But there is a little bonus that I put in here. Transcranial magnetic stimulation, TMS. TMS is a technology that lends itself to true experimentation in the field of neuroscience. All right, TMS focuses a strong magnetic field on a specific part of the brain. You can focus the magnetic field on different parts, and when the field is focused, those magnets will disrupt that part of the brain. You basically turn that part of the brain off temporarily. Transcranial magnetic stimulation appears to have no long-term negative consequences, so you better believe we're using it a lot. TMS temporarily disrupts parts of the brain so you can see what the area might be in charge of. This allows us to do true experiments, because we can manipulate the biology piece to see what changes occur in the behavior piece. And again, that's important. When it comes to doing experiments with TMS, we can manipulate the biology piece by disrupting certain parts of the brain, and then looking at what changes occur in the behavior piece, whatever function has been disrupted. And also, there is some therapeutic use to TMS. TMS is used in the treatment of major depression in the form of rTMS, which is rapid transcranial magnetic stimulation. Basically, with this, you just put on rapid pulses, rapid pulsation of transcranial magnetic stimulation. You have these magnets, and it's just boom, 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 boom. And that's therapy. All right, we're almost done. We just got to talk about the modular mind. We got to talk about research ethics. Modular mind, here we go. The modular mind is the idea that our brain is partly designed into modules that work together while also being independent. So, according to the modular mind, we have parts of the brain that are highly specialized to do stuff. Like, I would have a part of the brain that is totally specialized in all things hands, or I would have a part of the brain that is specialized in all things face. All right, that's the modular mind. Now, our brain is not entirely divided up like this, but the idea here is essentially modular mind. The mind has a certain organization that we can tap into with neuroscience approaches and imaging techniques. Just makes it easier to study the brain. All right, research ethics. We got this broken up into 3 parts. We're going to start with the cardinal rules, then we're going to talk about the safeguards, and then we're going to talk about the organizations. So, first and foremost, research ethics, cardinal rules. Plagiarism is the theft of another person's work or ideas. Plagiarism denies individuals the credit that they deserve, and it erodes trust among members of the research community. Now, to this point, you can actually plagiarize your own work, so if you get to writing a lot, just be careful. Then we have fabrication, all right, faking results. Fabrication is more serious than plagiarism, because fabrication introduces erroneous information into the body of scientific knowledge. That basically just means fake news. As a result of you introducing all this fake news into the body of scientific knowledge, the community starts pursuing these false leads, and pursuing these false leads consumes scarce resources and sidetracks researchers from actually pursuing more fruitful lines of research. You run these people on a wild goose chase by fabricating your stuff because you are making shit up, and these other researchers are like, oh wow, let's investigate that, not knowing that you're full of shit. Oh, and more importantly, fabrication in clinical research can slow therapeutic progress and potentially harm lives, because you gotta figure, if I start fabricating stuff and I'm like, oh, if you inject blueberries into the veins of people who have like histrionic personality disorder to freaking cure them, then it's like, oh yeah, sure, let's investigate that. Though yeah, that's going to slow therapeutic progress and it's going to harm a lot of people. So that is why fabrication is the worst right there. But of course, we can't forget about the last little cardinal rule here. We have deception. Deception violates informed consent. The three cardinal rules we've talked about are plagiarism, fabrication, and deception. Now, of these, deception can be considered acceptable, but only when we meet three pieces of criteria. When it comes to acceptable deception, the value of the study has to justify it. Alternative procedures cannot be available, all right? So there can be no alternative to using deception. And the individuals must be correctly informed about what they were fooled about afterwards. So if you use deception, you got to make sure that your study justifies it, you got to make sure that there was no other course of action, and you got to make sure that the people you deceived get told the truth afterwards. That's why you can use deception sometimes, but only when you meet those. Otherwise, deception violates informed consent. So in short, plagiarism is the thieving of other people's work, fabrication is making shit up, and then deception is you lying to participants and violating informed consent. Now let's talk about the safeguards. Here we get into ethics. Ethics are all about determining what is right and wrong to do professionally. Replication is the best safeguard against faulty research. Replication actually cures a lot of the ills of ethics violations in research. And to this point, the issue of people fabricating their results became such a big deal that this guy named Dr. Urey devised a statistical method to detect suspicious patterns in research data. I don't know if that's going to be on the assessments or anything, but that's just a thing. So the best remedy for issues in the area of ethics in research is replication. If you want to keep your ethics straight, use replication. However, that being said, there is an issue with replication in psychology. In the field of psychology, we see that it is actually very difficult to replicate a lot of famous experiments. How much can we really rely on them if we cannot replicate them? So that's a big issue. Talking about informed consent, when it comes to informed consent, individuals voluntarily agree to participate after receiving information about any risks, discomfort, or other adverse effects that they may experience. We gotta tell people about the time commitment and what sort of disturbing stimuli they may encounter. And because of this informed consent, we also gotta mention that at any point during the study, the participant may elect to leave freely. No consequences. Another thing to consider are vulnerable populations. Okay, vulnerable populations are groups of people who researchers may want to study, but who may be difficult to study. Researchers who want to study individuals with mental challenges. You want to study them, but they may prove difficult to study, making them a vulnerable population. Or researchers may want to study children, but of course children, difficult to study. So vulnerable population. In these cases, in working with vulnerable populations, researchers get informed consent from the primary caregiver. That's important. Informed consent, we're talking about individuals voluntarily agreeing to participate after getting debriefed on all the things they may encounter. But then with vulnerable populations, all right, we got to get informed consent from the primary caregiver, because vulnerable populations are groups of people that we may want to study, but that may be difficult to study. Okay, last but not least, research ethics, talking about the organizations. I want you to know about the IRB, and I want you to know about IACUC. Starting with the IRB, the Institutional Review Board, okay? The IRB watches over the researchers. The IRB is used in both schools and in private sectors that do their own research. The Institutional Review Board is a committee that applies research ethics by reviewing the methods proposed for research to ensure that they are ethical. The IRB is basically a council of people who oversee you doing your thing to make sure you don't act a fool when you conduct research. We see this in both schools and in the private sector. Then we have IACUC, alright, the IACUC, Institutional Animal Care and Use Committee. The IACUC is basically the IRB of animal research. IACUC advocates for the ethical treatment of animals. And why would you use animals in research in the first place? Well, animals bred for research have 3 important advantages. For one, research animals live in a controlled environment that lends a lot to your experiment. Research animals have a homogeneous history of experience. And then research animals have a briefer development and overall shorter lifespan, so it's less responsibility for the researcher. Because of these advantages, animals are used a lot in research, but they're not always used very nicely, which is why activists have raised concerns about animal research. That's why we have animal care and use guidelines. These have been strengthened, and also IACUC has increased monitoring, so we see a lot more monitoring of animal research. Alright, real quick before we go, I'm going to jump back to the stains. I just want to give a shorthand for this. Okay, so Golgi stains highlight individual neurons. Myelin stains emphasize white matter, and they let us see neural pathways. Nissl stains emphasize the cell bodies of neurons. These let us see the dendrites and the somas, or gray matter. And then Ch.4 stains mark the nuclei of active neurons. So if we want to see the nuclei of active neurons, we use Ch.4 stains. All right, that's going to do it. Everything for research designs in Chapter 4 of Neuroscience. It's been fun, and I'll see you next time. Class dismissed.